As we discuss the process of gametogenesis, it is almost the same for a male or a female. So may it be we are talking about the primary spermatocyte or maybe the primary oocyte. We are beginning with diploid status, that is 2n. In this, the small n represents the number of chromosomes, and as such we are diploid organisms. We are having 46 chromosomes, and that 46 number is represented by 2n. In that 46 chromosomes, we are having 23 from our mother, that is the maternal chromosomes, and we are having 23 from our father, that is the paternal chromosomes. So we are getting 1n from mother and 1n from father. Now, since it is difficult to represent all the 46 chromosome in a cell, we are just showing one pair of chromosome, and it is understood they are homologous chromosomes. Like that, there will be 23 pair of chromosomes, making it total of 46. So here we can say, let us assume this is the paternal chromosome and that is the maternal chromosome. And like any cell in the body, may it be the blood cell, bone cell, muscle cell, hair cell, all these cells in our body, they are supposed to be 2n or diploid in status. So it is the same for the primary spermatocyte or maybe primary oocyte. Now, as the cell is undergoing meiosis, meiosis is two-stage process, meiosis 1 and meiosis 2. Meiosis 1 is the reduction division. It will reduce the chromosome number to half. As you can see now, during the first meiosis, we are having reduction of chromosome number since the paternal and maternal chromosomes, they are separating. Then the next cell, which is called as the secondary gametocyte, may it be the secondary spermatocyte or secondary oocyte, it is going to be haploid. So this is one haploid cell and that is also one haploid cell. As I suggested, n is the number of chromosomes and can be counted by the number of centromeres. Like here, we had 2 centromeres, so it is 2n. But here we are having now only 1 centromere, so it is 1. After that, the meiosis 2 will begin. And in meiosis 2, what we find is the sister chromatids are going to separate. There will be a break at the centromere and the sister chromatids are not separating, as is the same seen here as well. So meiosis 2 is just like mitosis. It does not change the chromosome number. It is the meiosis 1, which is a reduction division, reduces the chromosome number to half. But here, as the number of chromosome is counted by the number of centromere, we have only 1 centromere here, so it is having n number of chromosomes, or you can say 23 number of chromosomes. So we are having 4 gametes now, and they are ready for fertilization. May it be male or female, as I suggested, they undergo the same process. So we will find that the sperm is having the haploid number of cell and is going to meet with the oocyte, which again is coming from another individual but having 23 chromosomes, that is the haploid status. And when the oocyte is fertilized by the spermatocyte, we find they'll form what is called as the zygote, and the diploid status will be then established. And then this diploid is going to form all the cells in the body. May it be the blood cell, skin cell, hair cell, muscle cell, or even the primary gametocytes. As I suggested, they will be diploid status, half the number of chromosome coming from the sperm, that is the paternal side, and half the chromosome coming from the maternal side. So you can understand the reason why there is reduction division and why should we make the chromosome number half, because eventually each gamete should have only half the chromosome number, and as fertilization occurs, diploid status is re-established. Now, as we proceed further, we can start with the zygote here. As we have mentioned, the zygote is having a diploid status. And here we can also mention it is not only 2n but also 2n. To explain the detail, the small n is the number of chromosomes, as we have mentioned earlier, whereas the capital N is the amount of DNA. So it is understood that 1 unit of DNA will be brought by the maternal side and 1 unit of DNA will be brought by the paternal side. You can see here we are having 1 maternal nucleus and 1 paternal nucleus, each of them bringing N and N. So together the zygote will be having 2N and 2N designation. It is understood that there are total 46 chromosomes in any cell which will be derived from the zygote, and in those 46 chromosomes, 23 would have come from the maternal side and 23 would have come from the paternal side, making it total 46. This is just a way of representing the number of chromosomes and the amount of DNA by the designation 2n and 2n. Now, as the zygote is dividing continuously, it is going to form what is called as the blastocyst. In the blastocyst will have inner cell mass, and in the inner cell mass will having the epiblast cells. These epiblast cells are the one which will be then going to form the primordial germ cells, and primordial germ cells are the first sex cell of our body. They have the capacity to form either the male or female gametes. If an individual is having Y chromosomes, then the primordial germ cell is going to form the sperms, and if we do not have the Y chromosomes, the same primordial germ cell is going to form the oocytes. So depending upon the presence or absence of the Y chromosomes, specifically the SRY gene on the short arm of Y chromosome, which is acrocentric chromosome. So Y chromosome is acrocentric, and on the short arm we have one gene which is the SRY gene, sex region on the Y chromosome. This is the one which will decide whether we are going to have testes and the sperms, and if we do not have the SRY gene or the Y chromosome, then we are going to have the oocytes. But both of them developing from the same cell, which is the primordial germ cell. This primordial germ cell undergoes mitosis to form the gonocytes, then the gonia. It may be the spermatogonia or the oogonia, which will then continue with mitosis and form primary oocyte if it is a female, or primary spermatocyte if it is a male. Primary cells will become the secondary cells, and later they form the gametes. If it is a male, then we'll find the secondary spermatocyte forming the spermatid, and then the spermatid is going to form the spermatozoa. So these are the spermatozoa. The examiner can ask you the sequence of gametogenesis, and you can mention first we were having the primordial germ cell, then we are having the spermatogonia, then primary secondary spermatocyte, then the spermatid, and finally the spermatozoa. But in case of a female who doesn't have the Y chromosome, we can tell the primordial germ cell is going to form the oogonium and then the primary secondary oocytes, and it is the secondary oocyte which is also called mature oocyte, which is ready for fertilization. Now one individual will give us the oocyte and the other individual will give us the spermatocyte, and when fertilization happens, there will be formation of the zygote. As we have mentioned, the oocyte will be bringing half the number of chromosome and 1 unit of DNA, whereas the sperm also will be bringing half the number of chromosomes and 1 unit of DNA. So as the fertilization occurs, we have 2n and 2n designation in the zygote, and the process then continues. Now as I mentioned, there is mitosis involved in the beginning and then meiosis involved. So we want to find out what is the difference between mitosis and meiosis in the next diagram. In this diagram, we are going to distinguish the mitosis cell division from the meiosis. It is understood that the mitosis is an equational division, means it is not going to change the chromosome number, whereas the meiosis is a reduction division. That means it is going to change the chromosome number. It will reduce the chromosome number into half. So how does it do? That we will see. But first, let us focus upon the mitosis. Now mitosis happens in all the somatic cells and partly even in the gamete cells, and you can think of any somatic cell, maybe the blood cell, muscle cell, skin cell, hair cell, which is going to have 2n chromosome status, where the small n is the number of chromosomes and the capital N is the amount of DNA. Though we are having 46 chromosomes in the cell, we are representing only one pair of chromosome here. We'll have 23 pair of chromosomes total, one from the maternal side, one from the paternal side. These are the homologous chromosomes seen here, and this chromosome status, that is 2n and 2n, does not change in mitosis. As you will see, even the daughter cells here, they will have the same status 2n and 2n. So as we are mentioning, the daughter cells are having the same chromosome and DNA designation as the parent cell. Now, before any cell division occurs, there is interphase, the resting phase, and in interphase there is S phase, the synthetic phase. That S phase, the synthetic phase in the interphase, is important because there will be DNA duplication here. Before the cell divides, there must be DNA duplication. So here you can see, because of the synthetic phase of interphase, there has been some DNA duplication, and now what we get are sister chromatids, and each sister chromatid having 1 unit of DNA. So if you talk about the genetic designation here, we are going to have 2n and 4n. As I have mentioned, number of chromosomes can be calculated by the number of centromeres. There are only 2 centromeres, so 2n, but the amount of DNA is 4n because each sister chromatid carries 1 unit of DNA. So 1, 2, 3 and 4. 4 sister chromatids having 4 units of DNA. Or simply, as you can see, the chromosome number has not changed, but the amount of DNA is doubled. In the S phase, there is DNA duplication and the amount of DNA doubles. The 2n becomes 4n now. Then as we understand, mitosis will have prophase, metaphase, anaphase and telophase. You can mention here, mitosis is going to have 4 subdivisions, that is the prophase, metaphase, anaphase, telophase, and it is the anaphase where the sister chromatids, they'll be arranged at the equator, there'll be a break in the centromere and the sister chromatids will then separate. So as there is a break in the centromere, one sister chromatid from the maternal side and one sister chromatid from the paternal side will move to the daughter cell, and similarly one from maternal and one from paternal side move to other daughter cell. Eventually what you get is 2n and 2n, which is the same as the parent cell, 2n, 2n. So mitosis is an equational division, happens in somatic cells mostly, and does not change the chromosome number. It stays the same. But that is not the same for meiosis. Meiosis is a reduction division, and it is going to reduce the chromosome number to half. The eventual cells, which are called as the gametes, maybe it is the sperm or maybe it is the oocyte, depending upon whether you have the Y chromosome or not. These gametes are actually haploid. The chromosome status is N and N, which is essential because then only when there is a fertilization happening, the sperm will bring NN and the oocyte will bring the other NN and the diploid status will be restored. So the daughter cells here are haploid when you talk about the meiosis, whereas the daughter cells in mitosis were diploid. Now let us focus upon meiosis in detail. So here we are beginning with what is called as the primary gametocyte. This could be a male or female. Depending upon that, you can say it is primary oocyte or primary spermatocyte. And the chromosome status for any normal cell in our body is 2n, 2n. Small n is the number of chromosome and capital N is the amount of DNA. And we are having 1 unit of DNA from mother and 1 from father. Now, before any cell division, we are having interphase, the S phase, synthetic phase. And during that, there will be DNA duplication. So as the cell is having DNA duplication in the synthetic phase of interphase, you can see now we are having sister chromatids, and that 2n, 2n has now become 2n, 4n. So you can mention here 2n and 4n. The number of chromosomes do not change, but the amount of DNA is doubled. As we understand, counting off the number of chromosome is by counting of centromere. We are having 2 centromeres, so 2n, but we are having 4 sister chromatids, each having 1 unit of DNA, hence 4 units of DNA, 2n, 4n. Now meiosis is 2-stage process, meiosis 1 and meiosis 2. And in meiosis 1, we are further having substages, that is prophase 1, metaphase 1, anaphase 1, and we are having telophase 1. Then further in prophase 1, we are having subdivisions, which are leptotene, zygotene, pachytene and diplotene. So leptotene, zygotene, pachytene and diplotene. And in pachytene, we remember a mnemonic PTC. So in pachytene of prophase 1, we are having a mnemonic PTC. P represents the pachytene, T is for tetrad and C is for chiasma. So P is for the pachytene, T is for the tetrad and C is for the chiasma. As you will notice now here in pachytene, there is a crossing over happening. The maternal and paternal chromosomes, they come together, and the non-sister chromatids, they exchange the genetic material. Now, since we can count 4 chromatids arranged together, that is why the name tetrad is there. And since there is some crossing over happening, exchange of genetic material, that is why the name is chiasma. At the chiasma, there will be some exchange of genetic material, and this genetic exchange happens only in meiosis. That is one difference between mitosis and meiosis, that the genetic exchange happens only in meiosis. As you can notice here, finally, maternal chromosome having a piece of paternal DNA segments or genes, and similarly the paternal chromosome having some DNA segments or genes from the maternal side. So interchange of genetic material. That happens during Pachytene stage of the prophase 1 of meiosis 1. And then in meiosis 1, the maternal and paternal chromosomes, they are separating. And finally, we are getting the secondary gametocyte. This secondary gametocyte could be secondary oocyte or secondary spermatocyte. And the genetic designation will be N and 2N. As I am suggesting, counting the number of centromere gives us the small n, and counting the sister chromatid, which is 2, gives us the amount of DNA. So N and 2N here, and the same for this cell. So during meiosis 1, we are having reduction of chromosome number to half, and after that there will be meiosis 2. In meiosis 2, we will see that there is break in the centromere and the sister chromatids are now separating. And as we finally get what is called as the gametes, they will become N and N. As now we have only one sister chromatid, and of course one centromere will have N and N designation. Gametes are haploid, and we need that because one gamete having NN, the sperm, the other gamete having NN, the oocyte, as they fertilize, the normal status, that is 2N, 2N, will be restored in the zygote. And in this diagram, we want to see the difference between the male and female gametogenesis. As we can see, in both the sexes, first we will have the primordial germ cell. May it be a female as shown in the diagram, or may it be a male. Primordial germ cell is the cell in the beginning to form the gamete. Now, continuing with the oogenesis or the female process, primordial germ cell will give us the oogonium, which will then give us the primary oocyte. Till this stage, we have to understand the cell division occurring is mitosis. So in the initial stages, there will be mitosis happening, which will convert primordial germ cell into oogonium into the primary spermatocyte or maybe primary oocyte. It is the same for a female or a male. The meiosis, that is the meiosis 1, begins only beyond this. So as you can see, first meiotic division, the cell undergoing first meiotic division is primary oocyte, and then we'll get what is called as the secondary oocyte. This secondary oocyte will then undergo meiosis 2, that is the second meiotic division, and we finally get the gametes. Now, one thing you can notice, some books call the gamete as ovum, but in fact it is the mature oocyte or the secondary oocyte ready for fertilization. But the other cells, as you can see, they are the polar bodies. This is the second polar body as observed here, and earlier we have seen the first polar body as well. Polar bodies are seen only in female, not in the males. And these polar bodies, they simply undergo apoptosis, they simply degenerate. So the ratio in a female is 1:1. You start with one cell, one primary oocyte, and eventually you are getting one gamete because rest of them are polar bodies. So ratio is 1:1. But for a male, it is 1:4. The males do not have polar bodies, so one primary spermatocyte will give you 4 sperms. The ratio becomes 1:4 versus 1:1 in a female. The other difference is, for a female, the process of meiosis 1 begins before birth, when the baby was still in utero. So when the baby girl is in uterus, the process has already begun. That is meiosis 1. But that is not so in case of a male. Remember, primordial germ cells, they remain dormant until puberty. So till puberty, there will be no meiosis happening in case of a male. Spermatogenesis begins only after puberty. So pre-pubertal testis will not have any meiosis. But as I am suggesting, pre-pubertal ovary will definitely be undergoing meiosis 1 because meiosis 1 in female begins even before birth in the intrauterine life. And there are some more details which we need for oogenesis, which can be discussed in the next diagram. So looking further in the process of oogenesis, in female, as we were talking about the primary oocyte, it would have entered meiosis 1 before birth in the intrauterine life only, but then this primary oocyte, which has already entered meiosis 1 before birth, is staying dormant in the dictyotin, or what is called as the diplotin stage of prophase 1 in meiosis 1. You can remember one mnemonic here, P1P. That is telling us the first P is the primary oocyte. The primary oocyte is arrested in prophase 1 and that too till puberty. So P1P. The primary oocyte remains arrested in prophase 1 till puberty. PP1P comes as one of the MCQ. And in prophase 1 also in diplotin stage of prophase 1, which is also called dictyotin, and this cell will proceed further only after puberty because what it requires is LH surge. So once we have attained the puberty, we are having the LH surge, then only the primary oocyte will become the secondary oocyte. And along with that, there will be release of the first polar body. Now, this secondary oocyte, which is released, will be further arrested in metaphase 2 of meiosis 2. So as you see, the secondary oocyte is now arrested in metaphase 2 of meiosis 2 and will be undergoing ovulation. So a question comes, which cell is undergoing ovulation? And your answer is the cell which undergoes ovulation is secondary oocyte arrested in metaphase 2 of meiosis 2. And this is the same cell which will be waiting for fertilization. And since we understand fertilization is a rare event, most of the time there will be no sperm available, so the cell will degenerate and undergo menstruation. So again a question comes, which cell undergoes menstruation? It is the same cell. It is the same cell which is the secondary oocyte arrested in metaphase 2 of meiosis 2, which was undergoing ovulation. Rarely it may be fertilized, and most of the time, since it is not fertilized, will be undergoing menstruation. But all the three events, the cell which undergoes these events is secondary oocyte arrested in metaphase 2. Now let us say the rare event has happened, fertilization has happened. What do you get next? What we get next is the mature oocyte, and along with that there is release of second polar body. We have to remember second polar body is released only after fertilization. So after fertilization you will see the second polar body, otherwise you will not see it. Now second polar body is seen only after fertilization. What about the first polar body? You can say that the first polar body is seen after LH surge but before ovulation. So if we are talking about the first polar body, it will be seen only after LH surge but before ovulation. This information we will be discussing once again in the next coming diagram. This is the diagram which is showing us the concentration of LH hormone during the menstrual cycle. As we understand, the menstrual cycle is calculated from the first day of last menstrual period, and usually we will take it as 28-day cycle. In this 28-day cycle, it is the mid-cycle, day number 14, that we are going to have the event called as the ovulation. Now, as we follow the difference in the concentration of LH hormone, we find that the concentration is having a sudden rise just before ovulation, and this is what we are calling as LH surge. This LH surge will be happening approximately 36 hours before ovulation, and then it will be taking a peak. So when is the peak? As you see, the peak of LH hormone concentration is happening at around 12 hours before ovulation. So LH peak is happening approximately 12 hours before ovulation, and it is the same time when we will have the release of first polar body also. So when is the first polar body released? First polar body is released approximately 12 hours before ovulation. That is at the same time when we are having the LH peak. Then when is the second polar body released? The second polar body is released after fertilization. Now if we say second polar body is released after fertilization, Then when is the fertilization taking place? The fertilization should happen within 24 hours of the ovulation. So within 24 hours of the ovulation, there must be fertilization. The female is having a very narrow window period for getting fertilized. If there was ovulation happening on day number 14, by day number 15, she must be having the zygote. Otherwise, what happens is the secondary oocyte will be degenerated and menstruated by the end of the cycle, and she has to wait for the next cycle to be fertilized and get pregnant. So a narrow window period of only 24 hours post-ovulation. Then what about the males? A question comes when the sperms are ejaculated, they are capable of fertilization. For how long? And you can mention once the sperms are ejaculated, they are capable of fertilization only for 2 days, that is 48 hours. So that means if these sperms were ejaculated on day number 12 or 13 or 14, then only there is a chance of fertilization, because sperms, they are capable of fertilization only for 2 days after ejaculation. So for a male it is 48 hours and for a female it is just 24 hours post-ovulation. This is going to be the most fertile 3 days around the day of ovulation. If the sperms are ejaculated before that period, no chance of fertilization, or after that period, no chance of fertilization. Fertilization occurs only if this narrow window period, the most fertile 3 days, were followed. Actually, the lady is given a LH kit, and when the LH kit become positive, she is supposed to undergo copulation to increase the chance of fertilization. Now, in the next diagram, we'll be talking about ovulation, fertilization, and implantation as well. Now, in this diagram, what we find is the uterus where the conceptus will be undergoing implantation. Along with that, we can see the uterine tube and the ovary as well. From the ovary, we are having the release of secondary oocyte, which will be then fertilized in the ampulla of the uterine tube or fallopian tube. So, as we proceed further, this is the ovary, and in the ovary, you can also see the Graafian follicle. There will be a release of secondary oocyte undergoing ovulation, and this secondary oocyte is covered by a glycoprotein membrane, which is called as zona pellucida, attracting the sperm. Now, this secondary oocyte will be attracting the sperm and undergoing fertilization in the ampulla of the fallopian tube. And once a single sperm nucleus is inside, it is the zona pellucida which will be preventing polyspermy. The zona pellucida is also preventing abnormal implantation in the uterine tube, and implantation can happen only in the uterus. For that, the zona pellucida must be lost. So, zona pellucida is preventing not only polyspermy, but it is also preventing abnormal implantation in the fallopian tube. Now, this is day 1 of ovulation. When the fertilization should happen within 24 hours of the ovulation. And on day 1, we will have the zygote. Day 1, we have the zygote, and day 2, we will have the 2-cell stage. And day number 3, we are having the morula. Now, this morula is having multicellular stage, which could be 12 cell, 16 cell, 32 cell, 58 cell. But if a question comes, we should answer the morula is having 16 cell stage. That is day number 3. And on day number 4, there'll be advanced morula. So day number 4, this is the advanced morula which has entered the uterine cavity. What is the cell number? The cell number you can mention is more than 16 until 64. So day number 4, the advanced morula has entered the uterine cavity, will have more than 16 cell stage and could be as many as 64 cell. On day number 5, we'll have a structure which is called as the blastocyst, having some blast cell and a cyst-like cavity. And this blastocyst is still covered by zona pellucida. So we must lose the zona pellucida, which is lost on day number 5. Then only we'll have the implantation happening. Implantation is a week-long process and is starting on day number 5. And it will be completed on day number 12, post-ovulation, or you can say post-fertilization, because the day of ovulation is also the day of fertilization. Fertilization should occur within 24 hours of ovulation. So implantation occurs, taking a week-long duration, and it is starting day number 5 as the blastocyst is hatching out of zona pellucida and will be completed day number 12. But then some questions come, they need more precise answer. For that purpose, you can say day number 7 to 9 becomes more precise answer. Day number 7 to 9, post-ovulation or post-fertilization, the blastocyst is undergoing implantation into the endometrium of the uterus. Now, in the next diagram, we want to look at the morula and the blastocyst undergoing implantation in more detail. And now, as we follow the morula entering the uterine cavity, changing into blastocyst, which is getting implanted here, we have magnified this picture of the uterine cavity and the endometrium to show it here. So this is the endometrium, this is the advanced morula, and as we have shown, advanced morula will have more than 16 cells, could reach up to 64 cells. And this will be day number 4, post-ovulation or post-fertilization. Day number 5 will find that there is a structure formed which is called as blastocyst. Called so because it is having some blast cells and a cyst-like cavity, hence the name blastocyst. The blast cells, as you can see, are embryoblasts, which are the inner cell mass. The inner cell mass of embryoblast will be going to form the embryo, whereas the outer cell mass, the trophoblast, will form the placenta. Tropho refers to nutrition, so trophoblast will help us in formation of the placenta, whereas the embryoblast or the inner cell mass will contribute to the embryo. Now, as the week number 1 changes to week number 2, that is day number 6 to 8, transition zone, we'll find that these cells are dividing further into 2 types. Embryoblast will form 2 types and trophoblast will form 2 types. So what are the 2 types of trophoblast or embryoblast? We'll be seeing on the transition of week 1 to week 2, that is day number 6 to 8, what will happen. Actually, week number 2 is the week of 2, and we'll find that in the week number 2, we are having 2 trophoblast, 2 embryoblast, and 2 cavities also. So week 2 is the week of 2. There you can see now the embryoblast has changed itself into the epiblast and the hypoblast. As the name is suggesting, epiblast means the cell which is on the top and hypoblast the cell which is below that. And this epiblast, which is columnar, and hypoblast, which is cuboidal, both of them, they came from this embryoblast or the inner cell mass happening at day number 6 to 8. At the same time, we'll find the outer cell mass or the trophoblast will be giving 2 types of trophoblast. So, as you see, there will be a cytotrophoblast and then syncytiotrophoblast. As the name is suggesting, syncytiotrophoblast, the cells have lost their cell margin and there is a syncytium appearance. And this is the syncytiotrophoblast which is helping us in implantation of the blastocyst into the endometrium of the uterus, and the cells which still have intact cell membranes will be called as the cytotrophoblast. They are towards the conceptus. And this syncytiotrophoblast, cytotrophoblast, they will be forming finger-like processes which are called as the villi. Like there will be primary villus, secondary villus, and tertiary villus. In primary villus, we'll have a core of syncytiotrophoblast and cytotrophoblast, which later becomes secondary villus and tertiary villus. As we see in the placenta formation, trophoblasts are involved in placenta formation, whereas the embryoblasts were forming the embryo proper. And then there are 2 cavities also visible in the second week of development. There will be one amniotic cavity and one yolk sac cavity. So week of 2 is week of 2. We have 2 cavities, the amniotic cavity, yolk sac cavity. We have 2 embryoblast cells which are epiblast and the hypoblast, and we have 2 type of trophoblast cells which are called as syncytiotrophoblast and cytotrophoblast. Now these 2 cavities, ultrasonographically, when you do transvaginal ultrasonography in week number 2, you will get one sign which is the double bleb sign. Here you can notice intra uterine pregnancy, there is a gestational sac showing two cavities, the yolk sac cavity and the amniotic cavity. Between the two, we are having the embryonic disc with the epiblast and the hypoblast. So, double bleb sign is ultrasonographic picture seen in week number 2 when you do transvaginal ultrasonography. Suggestive of intrauterine pregnancy, there will be a gestational sac with the two cavities, amniotic cavity and the yolk sac cavity, sandwiching the embryonic disc where we will have the epiblast and the hypoblast. As we proceed further with the details of the developing baby, we already have mentioned that the week 2 we were having two cavities and two cell types coming from the embryoblast. The cell which is above, and the name suggests, is the epiblast, the columnar cell, and the cell which is below, the cuboidal cell, is the hypoblast, as the name is suggesting itself. Similarly, there is a dorsal cavity which is the amniotic cavity and a ventral cavity which is yolk sac cavity. So, at the roof of yolk sac cavity, we are having the cuboidal cells, the hypoblast, and at the floor of the amniotic cavity, we are having the columnar cells which are the epiblast cells. We have to remember that the embryo basically develops from the epiblast cells, and actually hypoblast cells will be gradually removed from the baby. They'll be replaced by the migratory cells which are epiblast cells. So how are these cells migrating? This was actually week 2, the week of 2. Now comes week number 3, or you can say by the end of day number 14. That is the junction of week number 2 and week number 3. As the week number 3 begins, there is proliferation of the epiblast cells to form what is called as primitive streak. So primitive streak is formed by the proliferation of the epiblast cells. You can compare this diagram by just having an orientation that you are in the amniotic cavity and you have slightly tilted the embryo and you're looking from the dorsal view. Embryo appears like a disc. This is the embryonic disc. So if you look in a cut section, the embryonic disc will appear like this. But in fact, it is a disc-shaped structure in the other view. So in the dorsal view from the amniotic cavity, you can see these are all columnar cells which are the epiblast cells. And these epiblast cells, they are now proliferating to form what is called as primitive streak. Now we need to remember, formation of primitive streak begins from the tail end, what is also called as the caudal end or the posterior end. For a developing baby, whatever is tail is caudal is posterior. So this is the posterior end. Primitive streak starts forming at the posterior end and migrates towards the head end. So this will be the head end, or you can say the cephalic, or you can say the anterior. So this is anterior and that is posterior. The proliferation of epiblast cells, formation of primitive streak begins at the end of day number 14, that is beginning of week number 3, and it is starting to form caudocephalic. The direction is caudocephalic, which you can show here as well. Say this is the caudal end and that is the cephalic end. So what we find is towards the caudal end, epiblast cells are now proliferating and they are forming what is called as the primitive streak. So epiblast cells proliferating to form the primitive streak, starting at the caudal end, moving towards the cephalic end. This primitive streak will then develop one primitive pit, and through the primitive pit, these epiblast cells, they start jumping and moving ventral. So as the epiblast cells are proliferating, forming primitive streak, there forms a primitive pit here, and they're jumping into this pit and going ventral. This movement, this migration is called as ingression. So epiblast cells are undergoing ingression, moving from dorsal to ventral, and they are replacing the hypoblast cells. As they remove the hypoblast cell, take their place, they'll be forming germ layers. The very first germ layer to form is endoderm. So the same thing you can see in the other diagram here is, first you can tell this is the dorsal cavity, the amniotic cavity at the floor. We were having the epiblast cell. This is the ventral cavity, the yolk sac cavity. At the roof, we were having the hypoblast cells, but those hypoblast cells have been removed. We are mentioning that there is proliferation of epiblast cells to form primitive streak, and as they are migrating ventral, they have replaced the hypoblast cells and themselves forming the first germ layer, the endoderm. So first germ layer to form is endoderm. It is formed by the epiblast cells who have removed the hypoblast cells out of the embryo. The hypoblast cells, they move out of the embryo and their place is taken by epiblast cells forming endoderm. So first germ layer to form is endoderm, second germ layer to form is the mesoderm, and the last layer to form is the ectoderm. So formation of the three germ layers in a sequence, endoderm, mesoderm, ectoderm, is called as gastrulation, and this gastrulation is happening in week number 3. So gastrulation is the process of formation of three germ layers in the 3rd week by the epiblast cell migration called as ingression. Now this is looking at the embryonic disc from one view, and this is looking at the embryonic disc from the other view. So all these epiblast cells which were there at the floor of amniotic cavity, they have migrated downward to form some disc-shaped structures. So the single disc, as we see here, would be changing into 3 discs: a dorsal disc, a middle disc, and a lower disc. The lower disc will be made up of the endoderm, the first germ layer to form. Middle disc is the mesoderm, the second germ layer to form, and the upper disc is made up of the ectoderm, the 3rd germ layer to form. So they are in the shape of a disc. This dorsal disc will be now ectoderm. If you remove this disc, below that will be mesoderm. If you remove that as well, then the lowermost will be the endoderm. So 3 discs are now formed by the same cell called as epiblast cell. And as we move further in describing the developing baby, we have sequence of diagrams which we are supposed to discuss. We have just described this part of the diagram. Let us now focus upon this part of the diagram as well. And later we can move on to the 3rd component. But first, let us focus upon the 2nd component in the next diagram. And as I have suggested, we are looking at the developing baby in 2 views. One, we are inside the amniotic cavity looking from the dorsal view, baby appearing like a disc in a sequential manner, whereas in the other view it is kind of a transverse section where the discs are appearing just like a layer of cells. So we already have described this part and we are moving on to the second part. As is understood, first we were having 2 discs made up of the dorsal epiblast and the ventral hypoblast, but that has now changed in the 3rd week, and in the 3rd week we'll have the 3 discs: the dorsal disc, which is the ectoderm, middle disc, which is the mesoderm, and the ventral disc, which is the endoderm. The same thing can be seen here as well. The dorsal most layer is the ectoderm. So here we are showing the ectoderm. Ectoderm is going to form the outermost covering of the body. So that ectoderm, as we see here, can be seen in this section and this section as well. It is a longitudinal section of the developing baby. This is the head end and that is the tail end. So the dorsal most layer is the ectoderm. The next layer is the mesoderm. So this is the mesoderm, and as you will notice, the mesoderm is already forming what is called as the cardiovascular tube. So cardiovascular tube is coming from the mesoderm. We can remember ectoderm is going to form the neural tube, the central nervous system, brain and spinal cord, that comes from the ectoderm, whereas the mesoderm is forming some tubes, like one of that is cardiovascular tube. And then we have the ventral most layer, that is the endoderm, and endoderm will also form some tubes. One of that is the gut tube. So endoderm is the ventral most, will form the gut tube. Middle is the mesoderm, will form the cardiovascular tube. Dorsal is the ectoderm, will form the neural tube. But the arrangement of these structures now keep changing. The head end is coming close to the tail end, which is called as the cephalocaudal folding. So there is a folding going on. The head comes close to the tail end, which will be evident in the next diagram. So as we were talking about the cephalocaudal folding, you can see this is the head, the cephalic region, that is the tail, the caudal region, and the head is coming close to the tail. This is called as cephalocaudal folding, and we have taken one longitudinal section. That same section is being shown here. We already have described here the ectoderm going to form neural tube, mesoderm going to form cardiovascular tube, endoderm going to form the gut tube. The same can be mentioned once again. Talking about the dorsal most layer, the ectoderm, it has formed the neural tube, and neural tube means it is the brain as shown here and the spinal cord as shown here. So this is the spinal cord. Now, brain and the spinal cord, they come from the neural tube, and neural tube comes from the ectoderm. So this was the ectodermal derivative, the neural tube. What about the mesoderm? The mesoderm layer is the middle layer, and we already have mentioned it already has started forming the heart tube, and this is the heart tube which is coming from the mesoderm. But the orientation of the tubes is now changing as there is cephalocaudal folding happening. Then we can also talk about the endoderm forming the gut tube. As we have seen, the ventralmost layer was the endoderm, and this endoderm is now going to form the gut tube, which is evident here, the foregut, and then we have got the midgut, and then we have got the hindgut as well. So this is the foregut, then we have the midgut, and then we are having the hindgut coming from the endoderm. Here we can also see the midgut giving one diverticulum, vitalo-intestinal duct entering the umbilical cord region, and the hindgut diverticulum, which is called allantois, entering the umbilical cord region. So hindgut diverticulum, allantois, and midgut diverticulum, vitalo-intestinal duct, they become content of the umbilical cord. Both of these are derived from the endoderm. In this diagram, we are going to talk about a third cavity, which is called as the intraembryonic coelomic cavity. Actually, we have seen two cavities in the second week of development. That is the dorsal amniotic cavity and the ventral yolk sac cavity. But then comes a third cavity, which is called as intraembryonic coelomic cavity. It can be shown here, developing in the mesoderm layer. And since it is a cavity which is coming around like this, so we are showing it in two components, but it is a single cavity. So dorsal is amniotic cavity, ventral is yolk sac cavity, and then there is a third cavity, which is the coelomic cavity. In this diagram, you can see the dorsal amniotic cavity, ventral yolk sac cavity, and this one is the third intraembryonic coelomic cavity. Along with that, we can also talk about the dorsal layer, which was the ectoderm, is evident here. Later, it is going to form the neural tube. Then we can see the middle layer, the mesoderm, which is going to form the cardiovascular tube later. It is in this mesoderm the cavity is developing, and it will be splitting the mesoderm also. Later we will see the mesoderm gets split into two layers because of this cavity. And then the ventralmost layer, which is observed here, is the endoderm. We have seen endoderm lines the roof of the yolk sac cavity, whereas the ectoderm is lying at the floor of the amniotic cavity, and the mesoderm is splitting to enclose the intraembryonic coelomic cavity. Now in this diagram, we are again starting from the same diagram which we have seen, mentioning that the ectoderm is lying at the floor of the amniotic cavity and is going to form the neural tube. What happens is, first we will find that there is a neural plate formation, and that neural plate is now going to form the neural groove, and then this neural groove is going to change into the neural tube, and this neural tube will later form the central nervous system, that is the brain and the spinal cord. So neural tube has come from the neural groove, neural plate, or the neuroectoderm. Then as we talk about the mesoderm, mesoderm is the middle layer, and from here we get one tube. That tube is called as the cardiovascular tube. So this is the cardiovascular tube which has come from the mesoderm. And if we are talking about the ventralmost layer, that is the endoderm. The endoderm, which is lying at the roof of the yolk sac cavity, is going to contribute to the gut tube. As you can see, this is lining the yolk sac cavity all around now, and gradually it will be contributing to the three parts of the gut tube. May it be the foregut, midgut, or the hindgut. So here we have got the gut tube being formed by the endoderm. So we have got three tubes, the ectoderm forming the neural tube, the mesoderm forming the heart tube, and the endoderm forming the gut tube. Then we were talking about the three cavities. There was a dorsal amniotic cavity, ventral yolk sac cavity, and a third cavity was developing, which was called as the intraembryonic coelomic cavity. That intraembryonic coelomic cavity can be seen here. splitting the mesoderm laterally. So this is the intra-embryonic coelomic cavity developing in the mesoderm, and as you can see, this intra-embryonic coelomic cavity is later going to contribute to other cavities. Like intra-embryonic coelomic cavity will form three cavities later, namely the pericardial cavity, the pleural cavity, and the peritoneal cavity. And in the next diagram, you can see intra-embryonic coelomic cavity actually forming the pericardial cavity. So this is the pericardial cavity, which is actually contributed by intra-embryonic coelomic cavity. So if it is around the heart, you will say pericardial cavity. If it is related with the lung, you will say pleural cavity. And if it is related with the abdominal contents, you will say peritoneal cavity. May it be peritoneal cavity, pleural cavity, or pericardial cavities, they all come from the intra-embryonic coelomic cavity. Now we are going to talk about the two membranes, one which develops towards the head end, the buccopharyngeal membrane, and the other which develops towards the tail end, the cloacal membrane, and also the formation of the notochord. In these diagrams, which we already have seen, in one view where the embryo appears like a disc, and the other view where we have taken a longitudinal section of the embryo. Now here you will notice that dorsal layer ectoderm is meeting with the ventral layer endoderm with no intervening mesoderm. Towards the cephalic end, this is called as the buccopharyngeal membrane, which is going to be the location of the future mouth. Whereas if you look at the fusion of ectoderm, endoderm towards the caudal end, there we will have the cloacal membrane, and that will be the future anus. So this is the cloacal membrane where we will have the future anus. We are going to put the food into the gut tube from the mouth, and as the food is digested, the fecal matter will be coming out from the gut tube through the anal opening. So a question comes about the cloacal membrane where we find no intervening mesoderm because there is a fusion of the outer ectoderm and inner endoderm with no intervening mesoderm. The same thing happens here at the buccopharyngeal membrane, the future mouth, which is fusion of the outer ectoderm and inner endoderm with no intervening mesoderm. With this idea, let us proceed further to draw this diagram of the primitive streak, talking about its component and the development of the cloacal membrane towards the caudal end, buccopharyngeal membrane towards the cranial end, and then also the formation of the notochord. And for that, we have to draw a diagram ourselves. So as we draw this diagram, we can mention that this is the disc-shaped embryo, and towards this side is the caudal end or the posterior end of the developing baby, whereas this is the cephalic or the anterior end of the baby. Now, when we were talking about the disc-shaped embryo, we mentioned there is a proliferation of the epiblast cell, and it begins at the caudal end of the baby by the end of the day number 14, and it is called primitive streak. So primitive streaks start forming from the caudal end towards the cephalic end, and it will have three components. There is a primitive groove, which we can show here, and then there is a primitive node, which can be called as the Hansen's node also, and we are having one pit here, which is called as the primitive pit. So these are the three components of the primitive streak, namely number 1, the primitive groove, number 2, the primitive node or Hansen's node, and number 3, the primitive pit. The same can be described in the other section as we have discussed earlier. So this is the transverse section of the embryo. Dorsal is the amniotic cavity, ventral is the yolk sac cavity, and we are having some columnar cells which were called as the epiblast cells, and we have mentioned the two ends. This is the caudal end, that is the cephalic end, and there is now proliferation of the epiblast cells starting from the caudal end moving towards the cephalic end, and this is going to form the primitive streak, the proliferation of epiblast cells. It is the same story which we are showing here. So we can also talk about the pit then. This is the pit, the primitive pit, and now the cells in the primitive streak, they start ingression, they start moving ventral. So we can mention here the columnar cells, which are the epiblast cells, they start jumping into this pit and go ventral. As they go ventral, the ingression during gastrulation, they'll be replacing the hypoblast cell and take their place to form the endoderm layer. So that endoderm layer you can show here. The epiblast cells, they have formed the endoderm layer here, the first germ layer to developing from the epiblast itself. And then as the epiblast cells keep migrating downward, doing the ingression, going ventral, they'll form the mesodermal layer also. So here we will find that mesodermal cells are appearing. They also develop from the epiblast. And later, the same epiblast cells, they'll be forming the dorsalmost layer, that is the ectoderm. So may it be the ectoderm or mesoderm or endoderm, all of them come from the same epiblast migrating in the primitive streak. And we have mentioned the dorsalmost disc will be now the ectoderm, and if you remove this disc, below this ectodermal disc will have another one, that will be the mesodermal disc, and if you remove that disc also, below that will be the endodermal disc. So there are three discs in dorsal to ventral appearance, and that the same thing is shown here. Now the point is, mesoderm is not present everywhere. As you will notice, endoderm is fusing with the ectoderm in the cephalic end to form what is called as the buccopharyngeal membrane. Here in this region, this is the region of buccopharyngeal membrane where there is fusion of ectoderm and endoderm, which can be shown here also, buccopharyngeal membrane towards the cephalic end, where you will have the fusion of the ectoderm and the endoderm, but no intervening mesoderm. And similarly, towards the caudal end, we'll find the endoderm is coming close to the ectoderm, and there is fusion of ectoderm endoderm towards the caudal end, forming a membrane which is called as the cloacal membrane. So even at the cloacal membrane, there is no intervening mesoderm. Mesoderm is not present at the cloacal membrane or at the buccopharyngeal membrane. So the same cloacal membrane can be shown here as well. On the caudal end, we are having a cloacal membrane, and this cloacal membrane is the fusion of ectoderm and the endoderm with no intervening mesoderm. This comes as a question. The cloacal membrane or the buccopharyngeal membrane, there is fusion of the ectoderm and the endoderm with no intervening mesoderm. Then we can also talk about the formation of notochord. You will find that some of the epiblast cells which were migrating from the primitive streak in the primitive pit in the axis of the embryo, they are going to form what is called as the axial mesoderm or what is called as the notochord. So this is going to form the notochord. The epiblast cells in the primitive streak, as they jump into the primitive pit, they are in the axial line and they are going to form the notochord. So notochord is extending from the primitive pit till the buccopharyngeal membrane. The same thing you can show in this diagram as well. These epiblast cells, as they were jumping into the primitive pit, they start running in the axial line towards the buccopharyngeal membrane, and they are the one which are going to form the notochord, which is also called as the axial mesoderm. And since it is mesodermal, you can see it will be contributed by this middle layer. Over the notochord, you will have the ectoderm, and under the notochord, you will have the endoderm. So this notochord will be covered by the ectoderm. If you remove the ectoderm, then you will have the notochord. And below the notochord, ventral to notochord, you will have the endoderm. As you can see here, notochord is dorsally lined by the ectoderm and ventrally lined by the endoderm. This is the axial mesoderm or the notochord. And notochord, the location is extending from the primitive pit till the buccopharyngeal membrane. Now comes the concept of procaudal plate as well. So where is the procaudal plate? Procaudal plate or precaudal plate develops just prior to notochord in this region. So this is the precaudal plate, which is before the notochord, which is cephalic to notochord but caudal to buccopharyngeal membrane. And procaudal plate will have all the three germ layers: the ectoderm, mesoderm, and endoderm. You can show the precaudal plate here as well. This is the region of precaudal plate. As the name is suggesting, precaudal plate is before the notochord, hence the name precaudal plate. But precaudal plate will have all the three germ layers. It will have the ectoderm, mesoderm, as well as endoderm. So what is the difference between precaudal plate and the buccopharyngeal membrane? The difference is buccopharyngeal membrane has no intervening mesoderm. Precaudal plate does have the intervening mesoderm. It has all the three germ layers. The buccopharyngeal membrane has only two germ layers, ectoderm fused with endoderm, but precaudal plate has all the three germ layers, and it is sandwiched between notochord and the buccopharyngeal membrane.